Good afternoon, students. Hope all of you are doing fine. And today, from your biology, we are going to start with molecular inheritance. We are going to discuss about this very important chapter from Unit 2. So, this is basically chapter number, after your genetics, this is your chapter number 2 that you have, and that is from Unit 2. Okay. Now, what do we study in this particular chapter? What things do we need to know in this particular chapter? So first, let's make it clear. First, we will know about the molecules of inheritance. Okay. Then we will look into the packaging of DNA into chromosome. Okay. Third, we will look into how we could actually establish that DNA, they are the actual genetic material, or in fact they are the most important molecules of inheritance. How did we come to this particular conclusion? So we'll look into some experimental evidences that we have. So we will look into experiments in the search of, let's write it like this, experiments related to the molecules of inheritance. Okay. Then we start with important properties related to these molecules, or in fact the central dogma. Okay. We start with the central dogma. And in under central dogma, we are supposed to discuss about three most important molecular epic events. The first being your replication, the second being your transcription, the third being your translation. Okay. So we'll look into the mechanisms. Now, as far as the syllabus is concerned, okay, we are supposed to look into all of these mechanisms in relation to prokaryote cells only. Yes, there are some differences in this particular events and processes when it comes to prokaryote and eukaryote. So we will eventually look into prokaryotes with in-depth details, with slight differences, whichever is there in eukaryote. So we'll also focus on that particular part. Okay. Now, after looking into the prokaryotes and eukaryotic replication, transcription, translation, then we will talk about the operand systems in this particular chapter. Operands. And two important operands we are supposed to study in this chapter. First is your lag operand and second is your tryptophan operand. So these are the two operands we are supposed to study. Lag operand first, tryptophan operand. And after we are done with this, we have some molecular diagnostic techniques that we have to look into. First being DNA profiling or DNA fingerprinting. So we will look into DNA profiling or DNA fingerprinting technique in details actually. And we will also look into some other techniques like northern blot, southern blot. Okay. So all this we are going to look into in the final part, and eventually we end this chapter discussing about HGP, that is Human Genome Project. So there are a lot of things to be covered, starting with the molecules of inheritance, then how they are packed into chromosomes, experiments which were involved in the search to establish DNA as the genetic material. Then we start with the core part of this chapter where we understand about replication, transcription, translation. Followed by this, we start discussing about the operand, right? And then the different techniques, and finally finishing it off with Human Genome Project. So let's begin, and we will bring up some important concepts right from class 11. That is from the chapter of biomolecules to understand this particular chapter, as because we will be talking about the molecules of inheritance. Now, if you look into some history, so which are the molecules who are basically involved? Uh, we have discussed in the chapter of genetics, if you remember, that something called genes are present, right? Now, where can these genes be present for an organism? And what are these? These are units of heredity. Every one of us knows this, right? So they can be present in two of the molecules. They can be present in two of the molecules. What are those? It could be the DNA, which is predominantly acting as the genetic material of each and every type of organism, predominantly. But that doesn't mean it is the only type of molecule that we have. Besides this, we can also have the RNA, right? So DNA as well as RNA, they fall under what category of molecules? We call them as nucleic acid. You remember this? Nucleic acid. This is a concept from class 11 that we have already studied. Now, how did we come to know that this DNA, this RNA, they are the basic genetic material? Who basically is the pioneer in the history of this particular branch of biology? So, the first name that you need to talk about is Friedrich Miescher here. Friedrich Miescher. Okay, Friedrich Miescher. Now, this Friedrich Miescher, what he did is basically he started isolating the nucleus materials. Okay, from where you know? It's from WBC. Where did he get the WBC from? He got it from the pus. Whose pus? It is the leftover cotton bandages. Okay, cottons, bandages which were left in the dustbin. Where were they left? In the dustbin of hospital. So from there, he basically started collecting the pus. You understand, right? Pus means what? These are basically sacrificed WBCs of our body. Now these WBCs, they contain nucleus. Every one of us knows. Now, Friedrich Miescher was, very keen to know what is present inside the nucleus. In fact, what he found is he found that some sort of acidic molecules are found in the nucleus, some sort of acidic molecules. And accordingly, he started naming them as nuclein. So what did he call? Nuclein is the term that he has basically used. So nuclein was basically used by Friedrich Miescher. Okay. Now, later on, he also mentioned that these nucleins are acidic by nature. Acidic by nature. So every one of us knows, right? If you look into DNA, what is the full form of the DNA? Deoxyribonucleic acid. If you look into RNA, what is the full form of RNA? Ribonucleic acid. So how did this particular thing come into existence? How the naming on all those things came? We have already studied in class 11. Now, right now we are looking into a different, depending on our previous class 11 knowledge, we are basically looking into a separate thing. We are looking into the applied part of this DNA and RNA. So they were acidic by nature. So Friedrich Miescher was the first person, okay, to give us this very important information. Later on, there were several scientists who started picking up different types of experiments. We will discuss about three most important experiments in this particular field. And we could discover, or in fact, we could come to a conclusion that there can be two different types of molecules which can act as molecules of inheritance. That means from the parents to the offspring, which molecule is basically carrying the informations? Either of these two. It could be the DNA or it could be the RNA. Out of these two, any one. But predominantly, if you look into majority of the living system, what we can see is the DNA, not actually the RNA. It is, this molecule is very restricted to certain viruses. We know that, and we are also suffering due to that. We know one retrovirus is basically hovering around, and due to which we are separated like this, right? So this is basically restricted to whom? The viruses. Beside this, virons. Some of the virons are there. Beside this, some prions are there. So these are parts which already we are done. We are interested in this molecule right now. Now, so this is the first thing that you need to know. Now, we need to know some important thing, that to become eligible for the molecule of inheritance, what characteristic a particular biomolecule always needs to possess. Let's look into those characters first. So first character that makes a particular molecule suitable to act as molecule of inheritance is it should have capacity to replicate. So that becomes the first property or first characteristic of a molecule. Second important thing that you can look into is not only replicate, you must be chemically stable. So chemical stability becomes a very important factor here again. Now when I say chemical stability, what does this mean? See, if you look into a cell, there are different types of molecules, different types of reactions which are occurring. And we know these reactions are basically called as metabolic reactions. So during this process, different types of ions, different types of oxidative radicals can get emitted, and these radicals can basically unstabilize your molecules, or in fact if they have a tendency to react with them, then what is going to happen? Obviously the information which is stored over there will not remain preserved. And how do you think, if such molecules are not able to preserve those informations, how do you think would you be able to pass it to the next generation? So this becomes a very important characteristic. Chemical stability confers a molecule. The greater the chemical stability, the more you have the tendency of preserving the information. So we will look into, out of these two, who is more stable and why is it more stable, that also we will discuss. And the third important thing is it should have the capacity to get transmitted from one generation, or in fact one cell to the other cell. Okay, so transmissibility is another important property. Miscibility becomes another important property. So it should be able to, so that's all you need to remember, very simple. So a particular molecule needs to have the property of replicating. What do you understand by replicating? You are one right now. You remember when a cell undergoes cell division process, what happens in you have the different phases, G1, S phase, you have studied about the G2 phase, M phase, you have studied about the different phases, right? Now what happens during the S phase? When a diploid cell starts going through the different stages of the cell cycle, during the S phase, what happens? The DNA duplicates. That means doubling of the DNA takes place. And this doubling of the DNA actually occurs by a process which is called as replication. So not only your DNA, even the RNAs are also capable of doing so. So that is why we have categorized these particular molecules under the category of molecules of inheritance. Clear? So till this much we got an idea. So the properties which a particular molecule always needs to have to show that they are the molecules of inheritance are these three important basic properties. So you must be chemically stable, you should have the ability to basically replicate, and you should be transmissible. Okay. Now after knowing about all these facts, now we first of all we are going to look into some important things about these two molecules, and then we start with the next important thing. So by now we understood that it is the DNA which is more chemically stable. So we have basically two molecules of inheritance. Either it could be the DNA or it could be the RNA, right? It could be the DNA or it could be the RNA. Now here we are basically going to discuss what is the DNA made of, what is the RNA made of first, because it is required. Until and unless you understand this, it's sort of like a revision of your class 11. Until and unless you understand this, you won't be able to understand the second point that I told you, chemical stability. So out of DNA and RNA, DNA is more chemically stable in comparison to RNA. Why is this? That is what we are actually going to look into. So you remember that DNA or RNA, they are basically nothing but polynucleotide, or in fact you can call them as polymer of nucleotide. Remember, polymer of what? Nucleotide. When I use these two terms, polymer means long chain of some monomeric units. Nucleotide are those monomers. Nucleotides are the monomers which gets attached and ultimately forms the polymer. Clear? So both are polymers of nucleotide. This is one thing that we understand. Now, what is a nucleotide and what are they basically made of? You also remember this, that all nucleotides are basically made of nucleoside. What? Nucleoside. Along with nucleoside, what is compulsory? It is the phosphate which is compulsory. What is compulsory? The phosphate is compulsory. So nucleoside plus phosphate is basically going to make your nucleotides. Clear? Now, what is this nucleoside made of? Again, we know this, that I am using the short forms here, nucleotide. For this NTD I have written, and for nucleoside, what I have written is NSD plus phosphate is P. Okay, so what is this NSD made of? It is basically made of two things. One is your sugar, and that sugar, you know it is called as ribose sugar. Remember, ribose sugar. And along with ribose sugar, what is present or what needs to be present is obviously the nitrogenous bases. This is where the things are going to change. Nitrogenous bases. This is where things are actually going to change. Nitrogenous bases which are present in DNA and RNA are completely different. Okay, there are a lot of differences in between that. So the nucleoside is basically made of these two things. The ribose sugar is the first one, and the second one is your nitrogenous base. Along with this, what are you supposed to add up? Is the phosphate ion. You are supposed to add up what? The phosphate ion. So ribose sugar, nitrogenous base plus phosphate ion together makes your nucleotide. And polymer of nucleotide basically makes your DNA and RNA. This is very simple. It's not that simple, although there is something that you need to understand here, where the difference lies. One becomes DNA and the other becomes RNA. Now first let's understand this ribose sugar. Okay, you remember from the chapter of biomolecules. Okay, 5-carbon sugar. This is the 5-carbon sugar, and say this is your CH2OH, right? This is your carbon number 1, 2, 3, 4, 5. Remember, right? Now if you look into this, this is having a hydroxide here, hydrogen here, hydroxide here, hydrogen here, hydroxide here, hydrogen here. Remember? So what do you call this? What is the name of this particular sugar? It is ribose sugar, or in fact you call this as alpha ribose. Sorry, it's actually the beta ribose, not the alpha ribose. It's the beta ribose, because the hydroxide in the first position, that is the epimeric position, is basically on the top above the plane. So this is your ribose sugar, and this sugar is actually present where? Do you know? It's on your RNA, not on your DNA. Why, sir? There is some difference. Where is the difference? It is here, and you know this, that the second carbon, instead of holding a hydroxide, what happens is it simply holds a hydrogen. So what is missing? Oxygen is missing. So what has been removed? Oxygen has been removed, and that is why we have given the term deoxy. What is the name of the sugar? Ribose. So deoxyribose. Okay. And where do you find this molecule? Obviously in the nucleus. Remember? Nuclein, nucleic acid. Okay, these terms and terminologies were being used long back. So as you find them inside the nucleus and they are acidic by nature, so this is how the naming of DNA has come. So deoxyribo nucleic acid. This is how we actually go about it. Clear? That is how the naming of DNA has come into existence. Now, so this is your ribose sugar, what we have talked about. This is your second carbon. And in the second carbon, instead of holding a hydroxide, what is it holding? It is holding a hydrogen. So what is missing? Oxygen is missing. And that is why we call this as deoxyribose sugar. What is the name of the sugar right now? Deoxyribose sugar. This much is clear, absolutely clear. Deoxyribose sugar, what is this? It's very easy. Now, what happens is we need to look into the second part of it. What is the second part to make the nucleotide? That is the nitrogenous bases. And you know from your knowledge of class 11, nitrogenous bases can be grouped into how many categories? 2 categories. Do you remember? One category is what we call as purine category. The second category is what we call as pyrimidine category. Isn't it? And why do we call them nitrogenous bases, by the way? Because they are heterocyclic compounds and in their cycle you will find nitrogen along with carbon, right? That is the reason. So purine and pyrimidine, these are the two classes. And under purine, if you remember, what more do you have? It could be either adenine or it could be either guanine. Isn't it? Either adenine or guanine, which could be represented as A or represented as G. These things are well known to you, isn't it? So either adenine or guanine can be present under purine. Now under the pyrimidine, it could be actually 3 of them. It could be cytosine, represented by capital letter C, cytosine, and after cytosine we can actually talk about the thiamine, represented by T, or we can also add another that is called as uracil, which is actually your U. Got my point? So if you talk about nitrogenous bases, right, it can be grouped under 2 categories, purine, pyrimidine. Purine can be of 2 types, adenine, guanine. Okay, pyrimidine can be of 3 types, cytosine, thymine or thymidine. In certain books you will find thymidine written and uracil. Now, question is, are all the 5 different nitrogenous bases found in DNA? The answer is no, sir. It's no. Then which are the ones that we find in our DNA? And how are they basically attached to this sugar? That is our deoxyribose sugar. We know about all these things, right? So, in DNA, the nitrogenous bases which we can find, the purines are the same, adenine and guanine, purine, adenine and guanine. But the pyrimidines, if you talk about, they are thymine and cytosine. You don't find uracil here. But when it comes to RNA, have a look, we are actually differentiating in between the 2 molecules right now. So if you look into DNA and RNA, we can see these differences, right? Purine will remain same, unchanged, adenine and guanine would be found, but in case of pyrimidine, there is a change. What is that change? Instead of thymine, you find uracil and cytosine is very much present here. So this is the first difference that you can see. The second difference that you can note, here is if you look into the sugar molecule. So if you look into the sugar molecule, you will see that in the second carbon, what is missing in case of DNA? What is missing? Oxygen is missing. In case of RNA, it must be present. Take. So if you talk about the second carbon oxygen is absent in DNA. So we can put this as a point. Second carbon oxygen is absent in case of DNA, but it is present over here. So did you understand about this? So if you understood about this, you will see one interesting thing. And what is that interesting thing? That due to the absence of oxygen, by your knowledge of chemistry, every one of us knows that oxygen contains two lone pairs of electrons and it is highly electronegative. So it is going to invite whom? The positive ions come, come, come. Now if you are inviting the positive radicals or say positive ions, then what are you actually doing? You are becoming what? Chemically more unstable. Now, point to be noted, DNA doesn't have this oxygen. As because this oxygen is absent, second carbon oxygen, it's going to make a lot of difference in chemical stability. It will be more stable because there is no electronegative element over here. Getting the point? Absence of this electronegative element would ultimately result into what? Giving or conferring the molecule of DNA much more stability. Clear about the whole story? Understood? So, who is chemically more stable? If we want to go by this point, RNA or DNA? DNA becomes chemically more stable. Chemically more stable. Okay. Try to understand, even though in our cells, eukaryotic cells, we are having RNA, we are also having DNA. Then why is it that the RNA is not acting as the genetic material? It is only the DNA which is acting as the genetic material, and we are using the RNA for different other purpose. But why is it, if you are having both of them, RNA being a much simpler molecule, you are using the complicated molecule called DNA as the molecule for storing your information? The answer is very simple. It is chemically much more stable in comparison to others. The point is clear. So if you look into the point, this gives us some basic differences in between the DNA and the RNA, and which one is a better molecule, why is it a better molecule. Now let's come back to the point. Let's organize the DNA once again. Let's study first with the DNA. We will come to the RNA and their different types later. I'll actually use the concepts of your class 11 more of over here to explain the first part. Now please have a look about first, let's talk about the structure of the DNA. So if you are supposed to make the DNA, you will require what? Nucleotides. I already mentioned, because DNA is a polymer of nucleotide, and these nucleotides are basically what type of nucleotides? Deoxynucleotides. Why? Because the sugar is deoxy. As the sugar is deoxy, so the nucleotides accordingly becomes deoxy. Got my point? So deoxynucleotides are present. So how do you construct a deoxynucleotide? Let's look into the story. So this is your ribose sugar, and remember this is your first carbon hydroxide. Remember what is not absent here? Hydroxide is absent. This is your third carbon hydroxide, which is actually going to get linked with the next one, so it also doesn't exist. On the other side, let's talk about CH2OH, which is present here. So this is your fifth carbon, fourth carbon, third carbon, second carbon, first carbon. Okay. Now, if you look into this particular molecule, it is supposed to, this is your deoxyribose sugar, DRS. I have written it as DRS, deoxyribose sugar. It is supposed to get added up with what? Nitrogenous bases. Now, I told you it can be purine, it can be pyrimidine, either of this. It can be either purine or it could be either pyrimidine. Now, if you look into the purine, it could be either adenine or it could be either guanine. If you look into the pyrimidine, it could be either cytosine or it could be either thymine. Now, the moment you want to add the nitrogenous bases, you have to understand some basic things about it. So let me just get into the skeletal structure of this. So if you look into the skeletal structure of purine, which is actually 2-aminopurine. Okay, so you will have a ring-like structure that would actually look somewhat like this. Clear? Here you will have a double bond, and this is the basic skeletal structure of adenine and guanine. Okay, why is it basics? So see, you will find some differences like presence of an oxo group somewhere along with the presence of an H2 group, which makes it guanine, or simply having the NH2 group makes it adenine. So this is how the purines look like chemically if you look into their molecular structure. But simply if you talk about the pyrimidines will have a general structure which would not have this thing. This will be absent here. So if this is absent, that means what? It's a single ring structure. So when we talk about cytosine and thymine, it's a single ring structure. Now, the cytosine. Now, how do we number them? If we talk about the purines, how do we number them? How do we number them? This also is very important. You have to know this. The numbering of this actually starts from here 1, 2, 3, 4, 5, 6, 7, 8, and 9. Okay? Clear? Here you will also have a nitrogen, and this 9th nitrogen is always connected with a hydrogen. Here you will have a hydrogen. Okay? So this is all about the purine. Now let's talk about the pyrimidine. What is something that you need to know about the pyrimidine? So here you will have a nitrogen, here you will have a nitrogen, and this will remain connected to hydrogen. It's for sure for all the pyrimidines, That is, let it be cytosine or let it be thymine. Now, please understand one very important thing here. What I said, 9th nitrogen. So let me write it down over here so that you do not complicate this thing. So purines as well as pyrimidines they will get connected to ribose sugar in case of RNA or deoxyribose sugar in case of DNA. The numbers are important now. At carbon number 1, this is the place. This is the place, and the bond that would be formed is due to decondensation, okay, and removal of water. So how do we make it? See, this is a hydrogen. This is what? Your pyrimidine, isn't it? So this is your first. The numbering will actually start from here: first, second, third, fourth, and fifth and sixth. This is how we go about for the pyrimidine. So what happens is this hydrogen and this hydroxide, they will basically get liberated in the form of what? Water. This will get liberated in the form of water, and there will be a bond by which they will remain connected over here. So who can remain connected and how do they remain connected? So once it comes to purines and pyrimidines, let it be the deoxyribose sugar, let it be the ribose sugar, all of them will remain connected to carbon number 1. Carbon number 1. But purines remain connected. Purines. When it comes to purines, the purines will remain connected using what? It is the 9th nitrogen. So let me write it down. 9th nitrogen will be connected to first carbon here. Clear? First carbon kiska hoga? This is ribose sugar, and 9th nitrogen, it is obviously of which one? The purine. Got it? The connection is important. Now in case of pyrimidine, it will be first nitrogen, okay, connected to first carbon. Okay, so this is how you can connect them to either of the sugars. It could be deoxyribose sugar, it could be ribose sugar. Now let's say this is your deoxyribose sugar, and to this I have connected adenine. The moment we connect adenine, the name will change, and this together, that is the sugar plus the nitrogenous base, is supposed to be called as what? Nucleoside. You are supposed to call it as what? Nucleoside. And this nucleoside which has been made, the name will also undergo a change, and you will call it adenosine right now. You will call it what? Adenosine. Instead of adenine, you will be using the term which is called as adenosine. Clear? So once we talk about adenine, once we talk about guanine, once we talk about cytosine, once we talk about thymine. These are the 4 nitrogenous bases which can remain attached. So accordingly their names also undergoes change the moment they get added with the sugar. So it can be guanosine, okay, it could be cytosine, right, it could be thymosine. Clear? So have a look what has changed. SINE, SINE, SINE. This changes itself implies that it is nucleoside. So by looking into the name itself, you should be able to identify, is it a nucleoside or is it a nucleotide? Easy. Till this much everyone understood. So what did we construct till now? Nucleoside. What are we supposed to construct? We are supposed to construct deoxynucleotide, and for this we are supposed to attach another important thing. What was the name of the other thing? That is phosphate ion. So you have to add the phosphate. You also need to add what? The phosphate. So let's add the phosphate and let's see how the structure is supposed to look like and where is the phosphate supposed to get added up. So here is your deoxyribose sugar. Let me draw it. Okay. Here you have, say, adenine, nitrogenous base connected, first carbon. Here you are having hydrogen because hydroxide is absent. Here you are having hydroxide. This is your third carbon, right? Here you are having C. Let me just put it in this way. H here. So it becomes CH2. And what do you have here is hydroxide. Okay. What do you have here is your hydroxide. Now what happens is it actually remains attached to H3PO4, who is your well-known phosphoric acid. And if you look into the structure of phosphoric acid, it will actually look somewhat like this: hydroxide, hydroxide, and hydroxide. This is, look into this, H3, H3, there is a phosphate, oxygen 1, 2, 3, 4, H3PO4. Now this H3PO4, please understand, it's an acid and you have called it as a phosphoric acid. And we know when an acid, from the knowledge of our organic chemistry, we know this, that when an acid reacts with an alcohol, it is supposed to form what? Ester. So there should be a formation of ester bond. What type of bond? Ester bond. Now the name of the ester depends on the connecting atom. Okay, so accordingly you will call this as phosphoester bond because phosphate is basically going to remain connected. Let's see how it comes into existence. So here, let's say this is your phosphoric acid. This is your phosphoric acid, right? And what happens is this hydrogen can go out because acid, they usually have a tendency to donate their protons. So what will happen here? Hydrogen will go out, and once the hydrogen goes out, the hydroxide will basically interact, okay, and they will form this bond. So what do you call this bond as? What is this connecting bond called as? This connecting bond is called as, what is the connecting atom? Phosphate. What is the other connecting atom? Carbon, right? So what should be the name given to it? Phosphoester bond. So please remember, phosphoester bond. So I have a point to be made here. I want to make a point here, and the point is that your nitrogenous bases remains connected to your sugar by what type of bond? Glycosidic bond. What type of bond? Glycosidic bond. And your phosphate remains connected to the sugar using what type of bond? Phosphoester bond. Don't get confused, it's not phosphodiester. We did not come to that diester stage till now. It's the phosphoester bond. Till this much we understood. And what should you call this particular structure right now as? What should be the total name given to this particular structure, which is having an adenine, deoxyribose sugar, and a phosphate or phosphoric acid connected to it. You should call it as deoxy. Adenine is connected, so adenosine. Remember, the moment you connect it with sugar, it becomes adenosine. How many phosphates? Because all the hydrogens has the capacity to basically leave the oxygen and they basically become O minus, O minus. Remember, this is what we say. This ion, this hydrogen, because in aqueous medium, this is a mineral acid which can basically donate 3 protons, and it's a tribasic acid, we know from chemistry, right? So if you can donate, then you will automatically become what? PO4 3 minus. This is what we commonly know as phosphate, right? So exactly here also something similar is going to occur. The oxygens which are free, they are going to donate, they have actually donated their hydrogen and they have attained negative charges on them. Clear? Till this much we understood. Now let's complete the name. Let's come back. So deoxyadenosine. Okay. How many phosphate is connected? One phosphate is connected. So let me write it as monophosphate. How many phosphate? Single phosphate. So accordingly I have called it as monophosphate. Now please understand. Say what is this? Say your deoxyribose sugar. And to this what is connected? Adenine is connected on this side. And what is connected this side? Say phosphate is connected. Let me write it as first phosphate. To maximum there can be 3 phosphates which can remain connected. And the closest phosphate to the 5th carbon, this is your 5th carbon, carbon number 5, remember, carbon number 5 always holds the phosphate. So 5th carbon, this phosphate which is the closest one, you will be calling it as what? You will be calling it as alpha phosphate, beta phosphate and gamma phosphate. Okay. So if you are connecting 3 phosphates, what should be the name given to it? Instead of calling it deoxyadenosine monophosphate, you should be calling it what? Let me see. Write it down in your comment. Hurry up. Let me see. What is the answer? Come on, fast. Come on, write fast. I need the answer for this. What should be the name given to it? If we call this structure as deoxyadenosine monophosphate, then say to the same thing another 2 phosphate is connected, what should be the name given to it? Come on, hurry up, give me your answer. Let me see who can respond fast. No one? You are not able to make it out? Are you able to make out? What should be the name of this? Any answers? Anyone who is willing? Everyone is sleeping. Okay, D-adenosine Bugra, D-adenosine triphosphate. That is deoxyadenosine. Correct, Bugra? You have missed one thing here. It will be deoxy. For DE, we will write deoxy, right? So it will be deoxy adenosine monophosphate for this, then it should be called as deoxy adenosine triphosphate. Okay, so you are almost correct. You have missed just a simple thing. Now, we understood till this much that this is how we can also have DGTP, we can have DCTP, we can have DTTP, and obviously we can have DATP. Now, please understand, these are basically required for making a new DNA. But in DNA, you will never find that these molecules exist in the triphosphate form. Okay, yeah, Bugra, it's well done. You have tried, but others could also do it. Yeah, even Mr. Grammar Brazil, I do not know your name actually. It is much better if you use your names. Deoxyribonucleic, deoxyribonucleic. So sorry, sir. I have already mentioned here adenine here. You must see this. So you cannot use deoxyribo. So you simply use deoxyadenosine monophosphate. You are not supposed to use the ribo term anymore. The moment nitrogenous bases are present, you are not supposed to use that. Okay, so in this way we can have all of this. What should be the full form of this? Deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, deoxyhymidine triphosphate. So in this way, all of these, they are resources by which new DNA molecules will be made. So these are, in short, what? Your D and deoxynucleotides. And these deoxynucleotides are supposed to make your DNA. They are supposed to remain attached and make your DNA. So let's make the structure of the DNA. Now, who is the first person to give us the term DNA, not the structure, the term DNA? Who has basically given the first time the name DNA to DNA? Who used this term? The term actually has been used by Friedrich Mischer. Did not use it. Friedrich Mischer actually has done what? He has basically discovered that it is acidic by nature. But he could not identify the chemical structure of it. In fact, the name was basically given by Zacharias. Okay, Zacharias actually gave this particular name, but the structure was discovered in the year 1950 by Watson and Crick. Watson and Crick. So the structure was discovered by them, the name was used by him. Don't confuse these two things. Structure was discovered by whom? Structure was discovered by whom? Watson and Crick. In fact, there were a lot of propositions. Beside Watson and Crick, you also need to talk about a physicist, and that is Rosalind Franklin, who basically helped them, in fact, to develop the structure by her devised mechanism of X-ray crystallography. So how did they make it out, the structure of the DNA? The structure of DNA, how did they make it out? It is using the technique of X-ray crystallography. This was the technique which was used by them, Watson and Crick. Now, Watson and Crick model is also a very famous, name. It is also having a famous name, and that famous name to this particular model is the double helical model. Okay, the double helical model. So this is also better known as the double helical model, which is very popular. So in the year 1950, they were also awarded Nobel Prize later on for their discovery. But Rosalind Franklin did not receive it because she died. Okay, so this they have basically shared the Nobel Prize for the structure of the DNA. Now, let's quickly come back to understand whatever we have discussed till now. So we have discussed about the deoxynucleotides. You remember? Remember deoxynucleotides? Now, what are the different deoxynucleotides? That also you know. So let me make one deoxynucleotide to look into the double helical structure first. Okay, and then we discuss about the Watson-Crick DNA molecular structure. Okay. Yes. Uh, Now let's look into this. Say this is your deoxyribose sugar, and this is where you have adenine. This is where you have esterified phosphate. So this is your, how can we write it? Deoxyadenosine monophosphate. Correct? What do you have here? Hydrogen. What do you have here? Hydroxide. Correct? Now let's connect them to polymerize them. So here we can make another, and let's say you have guanine here, and let's say you are also having a hydroxide here, hydrogen here, and in addition to this, say you are also having phosphate here. Correct? So this is 5 prime, this is your 3 prime. Please understand, this is your 3rd carbon, this is your 5th carbon. Although over the phosphate I have written it, but it is connected to the 5th carbon. Now please understand what is going to happen. You remember that they are connected, the phosphates are basically connected in this way. In the previous chemical structure we have seen this. Remember? Yes or no? So what happens is again esterification is going to occur. So remember, this is your ester bond. So I did not draw it, simply represented it in the form of P. Now if you look into this, you will see a hydrogen going out, and again an ester bond has been formed. So tell me, how many ester bonds are basically connecting two nucleotides together? These are 2 nucleotides, isn't it? 2 deoxynucleotides. So how many ester bonds are connecting? Ester bond 1, ester bond 2. So what should be the name given to this ester bonds or the connecting link that acts as the backbone of the DNA? So you should be calling it as phospho, because it is the central atom here, phospho, di, because it is 2, and the bonds are supposed to be called as ester bond. So phosphodiester bond are the bonds basically that connects, or in fact the main skeletal structure which connects the nucleotides, or in fact, to be more correct, deoxynucleotides to each other. Now in this way, say if we keep on constructing, in this way, say this is another one, and this time I am not drawing the whole of the thing. So this is another ester bond, this is another ester bond, by which it is basically connected to the carbon, and say this is your structure that you can see. And in this way, if you keep on elongating the chain, say here you have cytosine. If you keep on elongating the chain, what can you see? What interesting thing can you see? Is where you started with the first nucleotide, where you started with and where you ended. Correct? So where you started with and where you ended, you can see a polarity has developed. What type of polarity? The first nucleotide phosphate, which is connected to the 5th carbon. It is connected to which carbon? 5th carbon. It is not bonded or esterified with any other nucleotide. It is not. So that is why we call this polarity as what? 5 prime polarity. Very important. 5 prime polarity. Exactly in the same way, say there are thousands of such nucleotides which are connected, by phosphodiester, 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 and the last nitrogenous base that will be there. In fact, the last nucleotide that you are going to find, this will always have the third carbon hydroxide free, isn't it? It will be free. So if it is free, this is a different polarity. So accordingly, we call this as 3' region. Now let me simplify this structure much, in a lucid way. Just have a look. Say this is your chain. This is your chain. And what should you have here? 5'. What should you have in this side? 3'. So two polarities, 5' to 3'. Now the direction 5' to 3', if you keep it constant, if you look into the Watson and Crick molecular structure, you will find they have two such strands and they are anti-parallel to each other, which means on the other strand, the 3' will be just opposite to the 5', and you will find the 5' over here. And both of these strands are basically connected by what? Hydrogen bonds. And who is basically supposed to connect to each other using these hydrogen bonds? That is what we are going to study in the Watson and Crick molecular structure. So please look into this. It's a double-stranded structure and it has anti-parallel nature. Anti-parallel. Now, I believe every one of you have seen railway tracks, right? So if you have seen railway tracks, do you see them meeting, the one track meeting with the other one ever? No, never, isn't it? So when we talk about this, that means there is a equidistant space which is always maintained throughout the whole of the track. So please understand this. When we talk about the Watson and Crick molecular structure, first point that you need to keep in your head is they have proposed that it is a helix. There is a difference in between helix and a spiral structure. And it is double-stranded. Double-stranded. And please understand, this is a right-handed helix. Right-handed helix, which means it is showing what? Clockwise fashion of turns. Clockwise fashion of turns. First point. Okay. So let's look into the structure. How is it going to look like? Say this is one strand. Then it is never supposed to. It is never, as it is a helix, it is never supposed to. Let me give you a very simple example to make it more clear. Say you have many of you might have seen this type of staircase, isn't it? Many of you might have seen this type of staircase. So if you talk about this type of staircase where you are basically moving like this, okay, so as it keeps on turning, the angle also keeps on changing, isn't it? And this is what we call a spiral. Now if you look into the railings are never supposed to, when you walk, you might have seen the railings, they are never supposed to intersect each other. They never intersect. And in fact, they are maintained at equal distance away from each other, right? So when we talk about the same helix structure in case of DNA, the same concept is also applied here. So let's consider this to be one of the strand, okay, which is going down like this. This is one strand. And let's say this to be the other strand, right? So please look into this particular thing, that there is no chances of intersection, and they have to be both of them should be following the same rule of turning. So please understand, in between this, what are you going to see? Base pairing. Now what is base pairing and what rule do we obey for this? That also we are going to discuss. So have a look into this. So this is how your molecules should look like, in fact. Now please understand, if you are looking into the same molecule from the top, these are the maximas. Maximas means what? Say you have started from here, you have come till here, so this is one point. Okay, this is the same another point. So these are the maximas of your, or in fact you can say, if it is a circular structure, what is this? Basically the circumference, correct? As if you look from the top, this will be the thing that you are going to find. So in a Watson and Crick model, what main features can we find? Is it is a right-handed helix which is having two strands, double strand. First point. Second point, the diameter of this is 20 angstrom. Can we remember this? 20 angstrom. Now say you started from here, you have come till here. So how much is this? And the moment you come over here, this is 90, isn't it? This is 90 degree, right? So from here to here, the change is obviously 90 degree, right? So from here to here, it's 180 degree, and coming back over here, it will be completing the whole of the story. That is 360 degrees. So that means starting from here, and if you come back till this particular point, you have basically completed, not till this particular point, it is actually till this particular point. Okay, one turn going and actually returning. So you have completed one helix. Now the length of one helix is actually 34 angstrom. So please remember the length is 34 angstrom. Okay. Now in this 34 angstrom, Watson and Crick, they have suggested that there could be maximum 10 base pairs. How many base pairs? 10 base pairs. How many base pairs? 10 base pairs can be there. So if there are 10 base pairs, say here you have adenine, here you have thymine, here you have guanine, here you have cytosine. Here you have guanine, here you have cytosine. Okay. Now what is the distance in between two consecutive base pairs? Consecutive base pairs. The distance in between the two consecutive base pair would be close to 3.4 angstrom. Okay. Now have a look into this particular thing. This area in the DNA is called as minor groove. What do we call it? Minor groove. And this is what we call as major groove. Major groove. So how do we define one helix? It's the distance from one minor groove to another minor groove, or one major groove to another major groove. This is a helix. Clear about it? Now, this type of DNA, later on after Watson and Crick, we have found out that DNA structures can be basically of 5 different types: A type, B type, C type, D type, okay, and Z type. 5 different types can be there. Now the Watson and Crick model is actually the B type, and I told you it is a right-handed helix. Now what does this right-handed helix means? Let me explain this using, I can demonstrate this, or in fact had it been inside the class, I could have shown you someone sitting in your classroom using, you might have seen girls having long hairs, right? So what happens is the intertwisting occurs. Now say these are the two strands. What you can do? Your right over the left, right-handed helix. Left over the right, left-handed helix. Right over the left, this is our B DNA, A DNA, C DNA, D DNA. All of these are right-handed helix. Z DNA is left-handed helix. Clear? So till this much we understood the structure of what? The DNA structure of which was basically given by Watson and Crick. Now in this there was further addition which was done by Chargaff, and this explains the base pairing rule. So what does the Chargaff's rule speak? So I'll make it very short and we will finish after this for today's class. So Chargaff's rule basically speaks about what? Speaks about the amount of purine and the amount of pyrimidine. Clear? amount of purine and the amount of pyrimidine present in the DNA is always equimolar. Always equimolar. So, purine concentration and pyrimidine concentration is always equimolar. So, what does this mean? Purines means adenine plus guanine. And what does the pyrimidine mean? Cytosine plus thymine. So, in a DNA, what is equal? Adenine plus guanine is equal to cytosine plus thymine. From here, we can basically come to this conclusion that this is equal to 1. This is equal to 1. Okay. Now, beside this, in the Watson and Crick model, they have also talked about the base pairing rule. They have also talked about, Watson and Crick have also talked about the base pairing rule, but they did not mention about this equimolar concentration of purine and pyrimidine present in the DNA. So, what did they say? That adenine always base pairs with thymine using double hydrogen bond, and guanine always pairs with cytosine using triple hydrogen bond. So, this is the base pairing basic rule. Okay. So, adenine always pairs with thymine with 2 hydrogen bonds and guanine with cytosine with triple hydrogen bond. So, this is the Shergaff's rule. Please remember this, and please remember the base pairing rules and the characteristic of the Watson and Crick model DNA structure. If you are having any confusions, you can write it down right now. I give you 2 minutes time and I can answer. Otherwise, we wind up for today. Any doubts, any confusion that you are having, you can ask it to me. Any doubts, any confusions that you are having, please do write in your comment box. इतने टैलेंटेड बच्चे हो तुम लोग, एक भी कोई क्वेरी नहीं है. Sir, yes, the last bond. Which last bond? Bugra? You're talking about this one, guanine and cytosine. Which one are you talking about? Is it this one, guanine plus cytosine? Guanine and cytosine, they are basically paired with triple hydrogen bond. They are with triple hydrogen bond. Okay, triple hydrogen bond would be there. Got it, Bugra? Only Bugra is having confusion. What about others sleeping? 3 or 2 something? Yes, sir. Yes, sir. It's 3. Adenine and thymine will be 2. Guanine and cytosine will be triple hydrogen bond. Okay. You have studied about hydrogen bond, sigma, pi, isn't it? So, accordingly 1 sigma, 1 pi here, and accordingly you will have the numbers of pi and sigma over in guanine and cytosine. Others, how many of you are there? 19 of you are there, right? Out of 19, I can see only one asking question. What about others? You are sleeping or what? See, this is a very important chapter where you have to clarify all the concepts, and if you try to memorize all these things, you will end up making a mess of whole of the story. Okay. So, if you are having any confusions, you can go through your text and you can keep your confusions ready. In our next day's class, we will start from here. Okay. And we will start with how basically the DNA packs to form the chromosome. So, thank you everyone for joining. Take care, stay safe, and please do go through your textbook. Thank you. Bye.